Hi, everyone. My name is Hillary Ruslow, and I have the distinct honor of serving as the Executive Director of the National Fragile X Foundation. And I just want to echo Eric and thanking you for joining us today. We're here together to discuss an update from Shionogi on their experience trials. My thanks to Shionogi and our colleagues at the FRAXA Research Foundation for joining together to create this really important opportunity. I think I speak for both the National Fragile X Foundation and FRAXA when I say that we so appreciate the thoughtful communication from Shionogi and their commitment to being present for the Fragile X community during these discussions. Thank you. As you'll see on the next slide, this webinar will be recorded and made available after today, so keep a lookout for those announcements. And it's lovely to be joined by Dr. Mike Tranfaglia, the medical director of the FRAXA Research Foundation, and Dr. Juan Carlos Gomez, who's the chief medical officer of Shionogi. On the next slide, we're going to just take a quick peek at the agenda. What you'll see is you'll hear from both myself and Mike, and then we'll get into the details from Shionogi. And then there'll be an opportunity to go through questions and answers. And our thanks to so many of you who submitted your questions ahead of time. Though all our questions might not be answered, I hope that we walk away from today with more understanding and certainly more hope for the future. Importantly, these trials would not have been possible without families' interest and willingness to participate in this research. This commitment cannot be emphasized enough. Because of you, there are new learnings for our community and progress being made. So thank you. It's my pleasure to introduce Dr. Mike Tranfaglia, Medical Director of the FRAXA Research Foundation, clinician, and perhaps most importantly, Fragile X dad. Mike.
Thanks, Hillary, and thanks again to all of you for tuning in. As many of you probably know, FRAXA has been heavily involved in the development of drugs that target PDE4 as potential treatments for Fragile X. We funded most of the translational research that established this target. We funded the preclinical research that validated zatolmilast as a therapeutic for Fragile X. And we funded the phase 2 trial with Dr. Barry Kravis, which was so encouraging. That is all to say that we're very heavily invested in this, as are all of you. And so we're looking forward to hearing more about the results of this trial and hoping to understand it a little bit better. And with that, I'll turn things over to Dr. Juan Carlos Gomez, the Chief Medical Officer of Shionogi. Juan Carlos.
Thank you. Thank you, Mike. Thank you, Hillary. Thanks to everybody for joining today and for your interest. And very particularly, of course, thanks to the National Fragile X Foundation and FRAXA for creating this forum to provide this important update on our development plan in Fragile X. Please, next slide. Okay, well, just a brief reminder, I think most of you know it, Shionogi is a mid-sized pharmaceutical company. We have our headquarters in Osaka in Japan, where the company was founded almost 150 years ago. But now we are very much a global company with large headquarters in the US, in New Jersey in particular, as well as other offices in the US and also a large European headquarters in London and in Amsterdam, besides our corporate headquarters that remain in Osaka. And as I said, we've really been, since our foundation, working in innovation in healthcare. Next slide. And in the last few years, we've got into the area of rare diseases, and in which, of course, our work in Fragile X belongs. And I think, as you already know well, pharmaceutical development and development of new treatments in rare diseases have significant inherent challenges, given precisely the small and dispersed patient populations. Frequently, the natural history of those conditions is not perfectly well defined. There is limited evidence. There are regulatory hurdles and obviously uh, issues with the study enrollment and logistics, given the limited number of potential participants. This is very true for Fragile X, as you know well. Besides, there's certain elements of Fragile X, as you all know, particularly the heterogeneity as well of the condition in which the phenotype, the symptoms vary enormously. It's a single genetic condition, but with many different presentations and obviously differences between genders and ages and between full mutation and mosaicism. So there is a significant heterogeneity, as well as there are really no well-established biomarkers yet. And given the fact that there's been no approved treatments for Fragile X, there are really no well-validated clinical endpoints yet, particularly no endpoints that have been fully accepted by regulators and that demonstrated that you can get an approval with them, given the inconsistent results that have happened in the last decade or more. Go to the next slide. So in this context of development in Fragile X, this very quickly a reminder about what is clinical development. I'm sure many of you already know this, but so you can situate what we will be talking about. I mean, as you know very well, it starts with a preclinical phase before going into humans. Then phase 1, which are the smallest studies looking at PK. Then we move into phase 2. In our particular case, phase 2 was the smallest study that Mike Tranfaglia referred to, the phase 2 study with satolmilast that was published in 2021. That showed very promising results done under the leadership of Dr. Barry Kravis at Rush University in Chicago. So this is the phase 2 study. And then based on that, we designed a phase 3 or phase 2-3 plan, if you will, consisting of two studies, one in adults and one in adolescents, and a long-term study. Then after the phase 3 trials, if those studies are positive and can create a submission dossier, then become the step of going to FDA for a regulatory submission. Let's go to the next slide. As part of this very brief reminder, as well, to make sure that we all understand each other, when we talk about endpoints, I refer to outcomes, measures that are used in a clinical trial to determine the effectiveness or safety of the drug being studied. So all the measures that are included in the clinical trial, the NIH toolbox or the ABC or the ADAMS, all these are endpoints. In every clinical trial, particularly in phase 3 clinical trials, there is always one endpoint or maximum 2 endpoints that are defined as the primary endpoint. This is is the measure in which the main hypothesis is based, is what we intend to demonstrate. We intend to demonstrate that the drug shows an effect compared to placebo, most likely, in a particular measure. That's the main hypothesis, and that's based on the primary endpoint. Then there are a number of other measures that constitute the secondary endpoints that are somewhat hierarchical, at a hierarchical level lower, below the primary endpoint. Some of these secondary endpoints may be predefined as key secondary endpoints. That means that, okay, they are the most important of the secondary endpoints, just below the primary endpoints. So we establish a certain hierarchy on how we are analyzing the results. And then I think I'm going to talk about signals, which this would suggest that is a study result that suggests that there is a treatment effect, or there could be a treatment effect, but may not be a conclusive result. For example, if a primary endpoint is positive, that is a pretty conclusive result, because that was your original hypothesis. You thought that you were expecting to see a result. That would be a conclusive result. If you see a result in other endpoints, that may be characterized more as a signal in which you see a positive result, but that not maybe entirely conclusive. Let's go to the next slide. So then going to the specific program, the experience clinical trial program with satolmilast, that was started originally by Tetra and then continued by Shionogi after our acquisition of Tetra. As has been mentioned, the phase 2 study was this study conducted at Rush University with 30 adult subjects with fragile X, all male adult subjects, in which 15 patients were treated with drug and 15 with placebo, and then switch in a crossover design. And particularly the first period, the comparison in the first period between the drug and placebo showed not only a very good safety profile, but also improvements across a number of the endpoints, which I will talk about them in subsequent slides. Then based on these results, then we design our phase 2-3 trials, the pivotal trials, which were essentially two. One that we called 204 in adolescents or older children and adolescents between the ages of 9 to 17. And then Experience 301, which was in adult males, 18 to 45. The primary endpoint on both trials, besides the age, they were very, very similar. There was a slight difference as well in the dose, because the younger children, 9 to 12, depending on the weight, they receive a slightly lower dose, which was weight-based. But besides that, the studies were designed almost identically, with the primary endpoint as the NIH Toolbox Crystallized Cognition Composite, which was an endpoint that had shown very good results in the phase 2 study, and then key secondary endpoints, which were the numerical rating scale assessing a caregiver assessment of language and communication, and then another numerical rating scale with caregiver assessments of daily function. And I'll talk about this a little bit later as well. And then all participants completing these double-blind trials, which were 13 weeks in duration, they could join into study 302, which was a long-term open-label, long-term extension study, primarily looking at safety and tolerability, but we certainly continued measuring all of the efficacy endpoints in the double-blind trials were continued to be assessed less frequently in the long-term study. One precision of course, in the double-blind trials, besides this primary endpoint and key secondary endpoints, there were also other secondary endpoints that I'm not going to talk about them too much today, but just be aware that there were other endpoints as well. Please go ahead. Okay, the NIH Toolbox, it's now, it's included in other studies in Fragile X, like Forward March, for example, is a cognitive instrument, which is a performance-based assessment. So this is not assessed by a caregiver or not assessed by a clinician. As most of you know, this is done by the participant themselves. So it's very relevant because it measures directly the cognitive performance, the intellectual performance across a number of tests of the person with fragile legs. It includes 7 different tests, but then 2 of them in particular, the picture vocabulary and oral reading recognition, that are really the verbal tests in the NIH toolbox, constitute this crystallized cognition composite. This was the measure that really performed very well in the phase 2 study. So we selected this particular composite to be the primary endpoint of these studies. In addition to having shown good results in that trial, it's a measure that investigators like David Hessel and others have shown that they are that they can be actually executed by almost all people with fragile legs. So at least down to 9 years of age, the majority of participants with fragile legs are able to complete the NIH Toolbox CCC proficiently. So this is very important, of course. Go to the next slide. Then these other key secondary endpoints that I have talked about, these numerical rating scales, these were an instrument that was really jointly developed by Shionogi with Dr. Barry Kravis and Rush University, based on other measures that had been used previously in other fragile X trials, that were visual analog scales measuring the worst problem as assessed by the caregiver. So the primary caregiver of a participant would select a primary problem and measure that through a visual analog scale. We changed that to be a numerical rating scale because that was preferred by FDA. And instead of only one problem, we decided to identify 3 problems and then take the average of the 3 problems by domain. So each caregiver would select what are the 3 most problematic symptoms or most problematic behaviors in language and communication and daily function. And then those would be assessed from a 0 to 10 scale. And language and communication and daily function were selected as the domains to be the key secondary endpoints. Of course, language and communication very much connected to the verbal measures from the CCC. And of course, the idea was that the improvement in language and communication would translate also in improvements in daily function, which of course are extremely relevant. Those also were measures that showed a positive effect in the phase 2 study. One thing, let me comment one thing. Both the NIH Toolbox as well as the NRS, they had not really been used largely in any other pivotal clinical trials for any other drugs. So in addition to including them as part of the measures of the trials, we also agreed with FDA in conducting a validation, so a psychometric validation, which is a study of the characteristics, the performance, the measurement performance of this instrument in parallel with the clinical trials. So let's go to the next slide. Okay, so then both trials, and I'll start getting into what we saw. Both trials, the adult study 301 as well as the adolescent study, went starting in 2022. So they progressed. Enrollment took several years, but they were running very much in parallel. Although the adult study was running a little bit ahead, approximately a couple of months ahead of the adolescent study. So, well, thanks to the participation of many of you and the help of the community, we completed enrollment for both trials one in April of 25, and the adolescent study in approximately in June of 25. So then we proceeded with the analysis of the adult study, which was the one that completed earlier. And we proceeded with the analysis as originally planned in the protocol and in the statistical analysis plan. And what we saw was that the NIH Toolbox CCC, the primary endpoint, was not met. Satolmilas did not separate from placebo. There was very much an overlap of the results between drug and placebo. On the other hand, there were, in the key secondary endpoints, there were significant improvements and relevant separation between the patients treated with drug and patients treated with placebo, both in the NRS for language and communication and daily function. But as I've said before, given that the primary endpoint was not met, these key secondary endpoints should be seen as signals, as kind of not fully conclusive results. In parallel with that, as I said, we conducted this endpoint validation that was planned and agreed with FDA how we were going to conduct it. The NIH CCC, we confirmed the already known property, so it had showed a good retest reliability and a good number of psychometric properties, but we could not confirm one of the key characteristics, which is the sensitivity to change or the responsiveness of the instrument. The CCC really did not correlate with any of the other clinical measures. Changes in the CCC, there were changes in the CCC in both directions, worsening and improvement, but those changes did not really correlate with the changes measured by the caregivers or by the clinicians. So ultimately, we could not confirm whether the CCC is actually responsive to change or not. In addition, the NRS, on the other hand, showed pretty good psychometric properties. Again, it was the first time that a formal validation was done, and I won't extend in details, but it showed pretty good psychometric properties and appeared to be sensitive to change. So when we found out these results, we were a few weeks before analyzing Experience 204, the adolescent study. It was about to complete, and we were going to proceed with the analysis. So at that point, we thought that, well, rather than progressing with the analysis as planned, with the CCC as primary endpoint and the NRS as key secondary endpoints, we thought that would lead to a negative study on its primary endpoint. We thought, well, if this instrument is really not sensitive to change, this may not be a good idea to proceed with the plan as originally intended. So we decided to approach FDA and discuss with them a switch of the endpoints. So we had to spend some time in requesting a meeting with FDA and meeting with FDA, because of course what we wanted is that all this was predefined. So before unblinding the study and going into the analysis, we wanted that to be aligned with FDA and not do it by ourselves. So we met with FDA and we got their alignment. Ultimately, it's the sponsor's decision, but we decided to proceed then with downgrading the CCC, so it was no longer the primary endpoint, and we defined the NRS language and communication and NRS daily function as the primary endpoints, as both of them as a family of primary endpoints. And then we added another key secondary endpoint, which was the clinician global impression of language and communication. In the same way as originally planned, this included a performance-based measure, the CCC, and caregiver assessments. We thought that it would be good to have a primary, which in this case was a caregiver assessment of the NRS, and then a clinician-based assessment with the CGI impression of change. Of course, the CCC was already measured. The study was done by this time. So, of course, we kept the CCC as a secondary endpoint. So it's not that that was eliminated at all. So let's go to the next slide. So then how did the trials look like? So then in the end, the way we have analyzed the trials was 301 as originally planned, with this primary endpoint, these key secondary endpoints, and then the adolescent study 204, then with the NRS as the primary endpoint and the CGI-I as for language and communication as the key secondary endpoint. No changes to the long-term extension study that kept as it was, with long-term safety, as well as a number of other efficacy measures measured over a long period of time. Go to the next slide. Okay, so we go then into a very brief summary of the main results, which some of them I've already shared, but I have here a summary of the main endpoints, primaries and key secondaries across both studies. So in the experience 301 in adults, the primary endpoint was the CCC, and that showed no significant difference between drug and placebo. Then the NRS, both for language and communication and daily function, showed a signal of significant improvement of drug versus placebo in adults. Similarly, another measure that we included, which was a caregiver global impression of improvement, which was similar to the NRS, but not specifically linked to any particular problem or behavior, a more global assessment, that also showed a signal of positive improvement. And then the clinician global impression of improvement for language, which was a key secondary in the adolescent study. In the adult study, didn't show significant improvement, although there was a trend. It really trended in a positive direction, but didn't reach significance. And then on the other hand, unfortunately, the adolescent study did not show any significant improvement across any of these endpoints. So the CCC did not show a difference, but also maybe more unexpectedly, the NRS for language and communication and daily function also did not show any differences, the same as the caregiver global impression or the clinician global impression. So in summary, neither trial met its primary endpoints. Several items in the adult trials showed positive signals of improvement. And then, very importantly, treatment with satolmilast was really very well tolerated. There were really no new safety concerns, very few discontinuations due to adverse events, very few discontinuations overall. The majority of participants joined, then followed into the open label extension, and overall a very good safety profile. Let's go to the next slide. So, okay, so what are the next steps from Shionogi? Well, I mean, we continue working on additional exploratory analysis of both the adult and the adolescent studies. So looking at, of course, at other endpoints, looking at subgroups, what we do data mining to really understand in in full depth the nature of the results that we obtain in the double-blind trials. And then, even more importantly, we still have pending the analysis of the open label extension study. This is, as I said, it's still ongoing. So we plan to do more of an interim data look. So we will do a database lock and an interim data cut in the coming weeks, so we can understand what the long-term data looks like. There are a significant number of participants that have been in the study for at least 1 year, and actually a very good number that have been in the study for 2 years. So we have a lot of long-term data. So we intend to look at that. At this time, the open label extension study remains open, at least until we conduct this analysis. So we didn't want to rush. We want to look at the data from the open label study until we make any further decisions. So at this point, participants who are already enrolled in the long-term extension study may remain if they wish to do so. And then we'll come back with updates at a later stage. For later part of this year, after we have this data from the long-term study, we will discuss with our clinical advisors, with the leadership of advocacy groups, and very likely we will talk with FDA before making any further decisions regarding the program. And then, of course, we plan to submit these findings, both the study results of the comparison of drug versus placebo, as well as the psychometric validation. We intend to present it at future scientific conferences, which are not fully determined yet, but we certainly will be presenting that at scientific conferences and certainly publishing them in manuscript format. I see there are some questions in the chat, but okay. I think I'm done. I don't know, Hilary, if there is any clarification from what you put in the chat that you think is necessary.
I think we're all set for now. We'll certainly move into Q&A shortly. Thank you, Juan Carlos. We really
Okay.
appreciate it. Thanks for walking us through things.
Okay. Yeah,
And
I realize how difficult it is to be presenting and then attending to the chat. So sorry,
yes.
sorry for that.
No, that's our job. That's what Mike and I are here for. So we're happy to be helpful. Yes.
Okay.
All right. Well, my thanks again. I think we appreciated getting sort of the high level overview and also really appreciated you outlining the next steps for us. That's helpful for our community and understanding what to expect and when we might be hearing from you. And so I just think it's important to acknowledge it's so nice when we get to be together, but in this case, we certainly wish we're under slightly different circumstances. And we want to acknowledge that many people in the community might be feeling frustrated or disheartened. And what we want you to know is remember there are so many things that we don't know, and that are being diligently explored as we speak and as Juan Carlos referenced. And in the meantime, remember that while there aren't specifically indicated treatments for Fragile X syndrome yet, there are certainly ways to treat those symptoms, from therapies to medications and combinations of those two things. There are lots of options for people to explore, and certainly dedicated professionals within the Fragile X Clinical and Research Consortium and beyond who are there to support you along the way. And, we are here too, the National Fragile X Foundation and the FRAXA Research Foundation. The NFXF is here to walk with you and provide you knowledge, resources, and tools in your day-to-day life until and even after effective treatments and a cure are achieved. There's just so much good science going on. There's policy shifts and guidances coming out, regulatory flexibility is being exercised, really innovative things coming out in terms of trial designs. And there's just so much work being done to bring about progress. So even though we have a ways to go, I know we can get there if we continue to work together. And we have so much work to do, but we really can't do it without you. So again, just my thanks to FRAXA for continuing to seed all these really important projects in the Fragile X community. And certainly our thanks to Shionogi for their commitment to communicating with our community and digging into the data. And most importantly, I just want to thank you for persevering. I know we will get across the finish line together. Mike, I imagine you might want to make some comments as well.
Thanks, Hillary, and thanks, Juan Carlos. I said this before, and I'll say it again. There are many, many reasons why clinical trials don't succeed. It's not just that the drug doesn't work. And I think a lot of the question and answer session will get at some of those potential reasons because you really do have to go through a process of troubleshooting and eliminating all these possibilities. But I think you need to keep that in mind that this was an almost but not quite. And that's really disappointing. And yet, in the midst of all that, I see some hopeful signs. And so I hope that everyone will persevere. Um, you know, we're getting our shots on goal, and one of them's going to go in one of these days. So I guess with that, we should go to the question and answer session.
Fabulous. All right. Well, thanks again to so many of you who submitted lots of questions, and we're going to do our very best to answer all of them. A ton of questions have also come in just now, and so please know we're collecting the questions and we're going to do our best to follow up with any additional information that comes up. But yes, please know that this is an ongoing effort and subject to change. All right. So let's dig into some of the details a little bit. So Juan Carlos, can you talk a little bit about why there was a positive effect in the phase 2 study, but the primary endpoints were not met in the phase 3 studies?
Yes, no, very relevant question. And I mean, here there is some important thing to say first, which is unfortunately, it's not uncommon that results from smaller phase 2 trials are not later confirmed in larger phase 3 trials. Approximately, 50% of programs you may see positive results in phase 2 trials that are later not confirmed. In our particular case, the phase 2 study was a relatively small study, as I said, with 15 patients on drug, 15 participants on placebo, which was provided very, very encouraging results in the NIH toolbox in these caregiver assessments. But then it was conducted only in one location, in one facility, a very good one. But then the moment in which you go to a significantly larger population with the heterogeneity that is inherent to Fragile X, as well as to other conditions, well, that's a risk that those results may not be confirmed. And it may be just a random effect that there was a certain group of subjects that showed a particular result, and then the moment you go into a larger population, that is not replicated. So there may be variability in the type of symptoms, the severity, the way the outcomes are measured across multiple individuals. In this particular case as well, that phase 2 study was only in adults. So now the phase 3, as you know, there was one study in adults and also the other in adolescents, which was the first study that had been done in adolescents. So maybe it's less surprising that there are not consistent results between that first study in adults and later in adolescents, since there may be some variability in terms of severity and evolution and interventions in adolescents and adults. So we clearly do not have a single explanation of why the small study showed results that have not been confirmed. I don't think we can say this is driven by this fact. This is probably a combination of many things, but mainly driven by heterogeneity.
That's really helpful. Thank you. Mike, I'm wondering if you have any comments you'd like to add?
I would just like to reinforce that that is the case in psychiatry, that a lot of the common drugs that we have in psychiatry have had large, high-profile failed trials and then gone on to get approval. So they're drugs that we use all the time and they definitely work, but they actually had failed trials. Like I said before, there are many, many reasons why a clinical trial might not be successful. And so sorting out those reasons, sorting through them and troubleshooting the trial process, I think is really important. And that's kind of where we are right now.
Thank you. All right, so we talked a little bit about endpoints. I want to continue that conversation. So as we reflect that, do you think that the endpoints used in phase three were the right ones to capture the meaningful change for people who are participating in the trial, the individuals with Fragile X syndrome?
Well, I think that with the information that we had at the time, I think it was the right choice. Um, those were endpoints that showed very promising results in the phase two study, both the CCC as well as the caregiver assessment, the personalized assessment of the major problem in language and communication and daily function. So I think based on what we knew at the time, I think it was a good choice. And as I said, the CCC is a performance based. I think that's been a long time desire in the community to measure directly, from the participants rather than through the caregivers or through the clinicians. Also, the caregiver assessments of language and communication and daily function, those are extremely relevant domains for the families in the Fragile X community. So I think they were the right endpoints. Of course, well, then you get results that are not what you expect, and then you may second guess. But my impression is that they were the right endpoints to measure something that was relevant that was relevant to caregivers, to people with Fragile X, and to clinicians.
I would
Thank you.
agree with that. I think that that was the obvious choice, and I'm not sure that it was wrong. I think that may not necessarily be the problem here, but we'll just have to figure that out by analyzing more of the data.
More work to be done. Wonderful. Well, let's see. As we continue to reflect, what about the time frame of the study? Was a 13-week study too short to observe any potential effects?
Um, I mean, that's also all questions are very relevant. This one is as well. Um, I think there was, again, with take into account that the phase three was really designed based on that phase two study, on what with the intention of replicating what that phase two study had shown. That was a 12-week period. So all the improvement that was seen in that trial was over a 12-week period. As well, I mean, 12, 13-week period is very, very common as a length of double-blind trials in many conditions, in many neuropsychological conditions. So I think it's a very reasonable time frame, particularly, as I said, with the intention of replicating what we had seen in phase two. Obviously, now we have longer term data, which we haven't looked yet, but well, we have it. We will be cleaning it and analyzing it over the next couple of months. So well, maybe the long-term data suggests that there are actually other efficacy signals that appear over a longer term. We don't know that yet. But I think in principle, the decision to do the double-blind trials 13 weeks was based on the evidence that we had at the time.
Sure.
Yeah. It will be really interesting to see the results from the open label extension. And personally, I'm expecting that the results would be better. But just to emphasize for everybody in the community, better isn't necessarily good enough, at least not for the FDA.
Good
Yes.
point.
Mm-hmm.
And what a testament to those families who've been in the open label trial for 2 years. That's quite a commitment. So I'm in deep admiration of that. All right. So again, thinking about the design and the trial details, do you think the trial dose may have been too low for some individuals?
Um, well, this is difficult to answer. I mean, let's say simply we think that the dose is the right dose. Um, but we have to admit that we haven't tested a higher dose. So I don't think we can definitely conclude that a higher dose would would have the same results. But the reality is that the dose was carefully selected at the time based on all the modeling from preclinical models, looking at what was the dose that is expected to exert its pharmacodynamic effect in the brain. And also, again, I keep coming back to the phase 2 study. That study showed the results it did with these 25 milligrams BID, and it's a dose that shows a very good tolerability, which of course is extremely important, I think, for participants with fragile legs as well as their families. So we think we had the dose right, but of course we cannot speculate. Would a higher dose have resulted in a different outcome? Well, the reality is we haven't tested that. So
Can I
I
just
think
ask
we
a,
have.
can I just ask a follow-up question, Juan Carlos? This seems like an area where further analysis of the data would be useful, specifically if you have drug levels to look at and try to compare rates of response with drug levels. Now, I know from participating in the trial with our son that the timing of the blood draws was sort of random following the last dose. So that's potentially an issue that complicates looking at everybody's blood level in the study. But it seems like it could be
Yep.
informative. Is that one of the things that you're analyzing as part of the?
It is definitely. Yes, yes. No, as you said, we have plasma levels. We will be doing concentration response analysis, which will inform this question as well.
So if you look at all the blood levels from everybody in the trial and the people in the top quarter of blood levels all responded beautifully, and the people in the bottom quarter, none of them responded at all compared to placebo, well, that does tell you
Yeah,
something, doesn't
yeah.
it? I,
That's definitely one of the things that we will be looking at.
I realize that you said that you'll be analyzing the data, but I think a lot of the people in the community don't really understand exactly what that kind of deep dive looks like. And so I know you guys are working on it,
Okay.
and we appreciate that.
Yeah, appreciate the deep dive. That's an interesting thing to know. All right, well, I'm going to try and get us through as many questions as possible. So let's see what we can do in the remaining time together. We spoke a little bit about the open label extension being open. Juan Carlos, can you share a little bit more about why Shionogi is continuing the open label study when neither of the two last studies met their primary
Yeah.
endpoints?
No, very, very important matter, and certainly something we've thought about. Um, well, first, it's important to realize these are separate studies. Of course, they are very much connected because 302 is an extension of the two other studies, but it's a separate study. And even the results of the two double-blind trials were not what we expected. We we decided that we don't want to terminate 302 before looking at the data, without looking at the data that we are collecting in this interim look, we think it would be premature to close that study. So we intend to use the study and deep dive into the data that we have collected and get a comprehensive understanding of the long-term safety, of course, but also efficacy, and look at whether are there any signals in the long-term follow-up of the participants in the study to fully understand also the results that we have already seen. So I think that's it. We think it's worthwhile. We will conduct this interim data look, and then based on that, we will make further decisions.
That's helpful. We've spoken about this a little bit. Both Mike and Juan Carlos, I think, have touched this, but let's just reiterate or perhaps go a little deeper. Um, when a phase 3 trial does not meet its primary endpoints, does that always mean that the drug cannot ultimately move forward and receive approval from FDA?
Um, well, of course, very relevant thing right now. Um, I, I think, I mean, we can say that the failure to meet the primary endpoint in a phase 3 clinical trial, it's a significant difficulty in any, regulatory process, obviously. But I would say as well that it doesn't entirely rule out a regulatory approval, particularly in rare diseases. I think there are
Hmm.
a number of rare disease conditions and treatments that have ultimately received FDA approval based more on the totality of data, despite maybe the main trial not meeting its primary endpoint. So again, I don't want to minimize the difficulty. So and again, I don't, we have to be very, very cautious here. I don't, I don't want to pretend anything, but yeah, let me be clear. It doesn't entirely rule out, but certainly makes it a difficult pathway for sure. And we certainly will continue, once we get more data from from the open label extension study, we will certainly engage with FDA and in determining any possible next steps with satolmilast.
Yeah, well, it's certainly a setback, but think of it this way. You can always do more trials. You're not prohibited from doing more trials. You're not prohibited in doing different kinds of trials. A number of companies, even in the Fragile X area, have proposed a number of, let's say, creative solutions to the FDA, and that hasn't gone over too well so far. In other words, post hoc analyses of data or trying to use different kinds of studies. So the FDA is pretty particular, but at the same time, it's not like one trial result dooms everything and there's no further development pathway that's possible.
Thank you. So I'm wondering, what would you most like families living with Fragile X to understand about these results and Shionogi's work within the Fragile X program?
Thank you. Yeah, this gives me the opportunity maybe to insist on maybe more or most important message is that I mean, clearly the results of the double-blind trials, the adult and adolescents, were not what we were expecting. Although we are still working on the full interpretation of both studies, so there is still more to learn, and particularly from the open label long-term extension study, which we haven't yet looked in any formal way. So we are still having to look at that. So we will come back in the latter part of the year with further updates once we have seen this data and be able to interpret it in full and maybe meeting with FDA. So we will come back with further updates later in the year. So more to come. In the meantime, let me, the other very important thing I would like to say is really extend our utmost gratitude to the individuals, the families, caregivers who have participated in the studies. And of course, as well, investigators, the site staff, um, the staff of the advocacy groups that have helped us as well in supporting these studies. These studies would not have been possible without you. So we really appreciate all the commitment of the families, the caregivers, and the participants themselves to uh, to the studies. It's really encouraging and heartwarming, the collaboration that we have had. Thank you for that.
So well said. All right. Well, finally, maybe a little bit of an altruistic question, but what can we do? What can the Fragile X community do? What should we do as participants during trials to ensure they're successful? That's what we all want. And are there things we shouldn't be doing or not? Can you just help us understand from your perspective?
Well, I mean, clearly, I think there is a very, very strong or very simple thing I could say, which is, please continue to participate in clinical trials. And I don't mean in Shionogi clinical trials, but in any clinical trial, I mean, in any good clinical trial. Please do that. That science cannot move forward without the engagement of the community and without the participation of the families and, of course, the people with Fragile X. I mean, there are certain elements in Fragile X. I think many programs have struggled with placebo response. So, be careful with that. Make sure that you follow the instructions of the investigators at the sites. Again, looking at the history of Fragile X, there are a number of trials in which large placebo effects have been problematic, especially related to subjective questionnaires by the caregivers. So make sure that you participate in trials and do so again following the instructions of the investigators at the sites where you are in. Be careful not to share, maybe too much in the community on your own experience. Try to, make your assessments based on your own personal experience. Try not to guess whether, if it's a double-blind trial, try not to guess whether your son or daughter is on drug or on placebo, and model your response based on your assumptions, but try to respond based on what is the real experience of you as a caregiver and your family member as a participant. Do not create expectations in the community, because that ultimately may hurt the clinical trials. Again, I'm not saying this was the problem with our clinical trials, but this is an issue in general. So keep participating in clinical trials. That is how science will progress, and do it in a as consistent as possible way, and in a very personal way.
So well said. Well, we're very close to time, so I just want to make sure both of you have the opportunity to close with any last comments. So perhaps Juan Carlos, please go first, and then Mike, you can conclude.
Okay. Yeah. Well, so thanks. I think I've said almost everything I had to say, but given the time, again, thanks everybody, particularly to the people with Fragile X who have participated, their caregiver, their families, and definitely the investigators and the site staff. And we'll hear from us. You will hear from us. We continue working, and we'll update you once we have any more news.
Thank you. Mike?
Keep the faith, guys. We've got the strongest patient community in the rare disease space. This trial was a great example of the kind of dedication and motivation of the Fragile X community. Shionogi needed 150 adults, they got over 170. They needed 150 adolescents, they got over 160. We're not done yet.
Wonderful place to end. Thank you. All right. Well, thank you again for joining us today. As you know, we mentioned the webinar is recorded and you'll receive notifications on follow-up shortly. And, just to reiterate our thanks for the many families who participated. We know it takes literal blood, sweat, and often tears to do these trials. And that is something that is so deeply appreciated by everybody in the Fragile X community. You're moving us forward to what Mike spoke to. So if you have, any questions, please feel free to email any of us. You can see our emails on these screens, and we're happy to be as responsive as we can. So thank you again for joining us, and we'll look forward to speaking soon. Take care.
Thank you, Hilary. Thank you, Mike. Thanks.
Thank you. Take care.